Good morning, everyone. My name is Diana Wall, and I am part of the Global Soil Biodiversity Initiative, and I'm really happy to welcome you today to our webinar. The Global Soil Biodiversity Initiative puts these webinars on once a month, and this one seems to be a very hot topic, so we're delighted to have you with us. I just want to give you a couple of things to note. All the questions and answers will be where you put your questions. Don't use the chat. The other thing is this will be recorded so you can see it on YouTube, and there'll be more information in the question and answer on that very shortly. And so I just want to, again, welcome you. And I'd like to introduce today's moderator. We're very fortunate to have Marcel van der Heijden from the University of Zurich, who does his research in collaboration with Agroscope. And if you don't know him, you're going to learn a lot about him now and read some of his papers. I think everybody may know him. And he will introduce the panel and we'll get started. Marcel?
Thank you very much, Diana. Good morning, good afternoon, and good evening, and maybe good night for some of you. It's my very big pleasure to moderate this session today on ecological networks. So as Diana said, I'm Marcel van der Heijden. I work at Agroscope, which is a Swiss research institute for agriculture, and I'm professor for agroecology and plant microbiome interactions at the University of Zurich. And so before we start with the four speakers, I will give a short introduction about the topic and about the importance of soil biodiversity in general. So as you know, the first half an hour we will give talks, and so if you have questions you can put them in the chat, but then afterwards, so there's no questions after the individual speakers, but afterwards at the end, so after half an hour, we will have a general discussion. So as you know, there's a lot to discover below ground. For many of us, maybe soil is dirt or something we walk on, but I think more and more research has shown it's really fascinating, it's inspiring. I think especially the research in the last 20, 30 years have shown there's so much going on below ground. So people have shown that about 1/4 of the total soil biodiversity is living below ground or living part of the life cycle below ground. So if you have a handful of soil, you probably know that if you teach about it, there are billions of bacteria, kilometers of fungal hyphae, and still a lot of unknown organisms we don't know about. So it's really a fascinating world. And it's actually very, very important for us human beings, because a lot of the food which is produced on soils. So why do we talk about soil networks? So soil organisms, they do not live alone. They are there with many, many different organisms, and they interact with each other. On the one hand, there are food webs. So where one organism predates another, eats each other, and then there's a top predator who again eats another organisms. But also many, many soil organisms, they associate with each other. They form mutualistic association. And for example, they do things which in other soil organisms cannot do. So they have also direct interactions. And then there are many indirect interactions, associations of soil organisms, and you will hear more about it from the speakers. So just a few examples. I have been eating some bread and had some rice in the afternoon. And we often don't realize when we eat food that the nutrients which are inside this food are actually delivered by soil organisms and by soil networks. So fungal hyphal networks, they forage in the soil for nitrogen, for phosphorus, for other elements which are important for plant growth. They give them to the plants so plants can grow. Much of the nitrogen which the plants get is processed by bacteria. So before it goes into the food, there is already a lot of stuff going on by these soil networks. And that's why it's very important to consider them, because they're very important for the food we eat. Maybe another example is water, drinking water. Drinking water should be clean, of course. We do not want it to be polluted, or if there's too much nitrogen, nitrate in the drinking water, because it's sometimes at very high concentrations toxic for young children. And so several researchers have shown that if you have complex networks of soil biota in the soil, they can filter nutrients. They keep them in the system. They keep them there for plants that they do not get lost. And so that keeps the water clean. So that's very important. So having good soil networks is really important for ecosystem functioning. Then what soil organisms also do, they can aggregate soil particles together. They glue them together with huge mycorrhizal networks, for instance, or fungal networks. They keep soil particles together. And why is that important? If there is soil erosion or there's heavy rain, the soil particles should stick together, otherwise they get lost and they cause problems. And also fertile agricultural soil gets lost. So these are important examples where bacteria, fungi form networks, they glue and bind soil particles together. And that's again important for soil quality. Then another issue, I think many of you are aware that climate change is ongoing, and we discuss a lot about carbon dioxide, trying to store carbon in the soil. And so again, also here, and I think this is an area where still much is to discover, soil networks can maybe help to sequester some carbon, or that it's not decomposed, but attached to soil particles. So I think there is a lot still to be discovered, and we will for sure hear much more about it in the coming years to come. So yeah, I would like to start to introduce the speakers. And now the first speaker will be Noah Fierer. I think many of you will know him. He's one of the pioneers working on microbial networks. He will give some very nice examples. He's working on it already for a long time. So it's a very big honor to welcome you, Noah, from the University of Colorado. So look forward to your talk.
Thank you, Marcel. Let me share my screen here. Okay, can everybody see that? Okay. So I'm a soil microbial ecologist. So I'm a bit biased in saying that I think it's a really exciting time to be studying the microbes living in soil and their contributions to ecosystems. This excitement is due in part to the widespread application of relatively new tools, including high throughput DNA sequencing and other omics tools that provide unprecedented insight into the microbial ecology beneath our feet, including the majority of soil microbes that remain undescribed. At the same time, the amount of data we can now generate is frankly intimidating and often overwhelming. So what do? We adopt approaches used by scientists in other fields that also have to wrestle with large data sets. One such approach is broadly termed network analyses. Now, network analyses have been used in all sorts of ways, from identifying foods that share flavor compounds in common with other foods, as shown here on the left, clearly garlic is king, to mapping collaborations between scientists at a research institute representing different disciplines, to describing marine food webs, the example on the right here. However, how we most commonly use network analyses in soil microbial ecology is a bit different. So let me explain using a simple example. So this example does not apply to all the ways network analyses can be used, just one way it's commonly used in soil microbial ecology. So let's assume we have a community with 6 taxa in it. Here, each taxon, or node in network terminology, has a different color. Now let's assume we did some meta barcoding, for example, of 24 soils to look at what's there using DNA sequencing tools. And we find a correlation between the abundance of sequence reads from taxon 1 and the abundance of sequence reads from taxon 2. You can tell this isn't real data because ecological data never really looks this good. Since the abundances of these 2 taxa are well correlated, they appear to strongly co-occur, so we will draw a line between them. Now let's look at another pair of taxa, and they are also correlated with one another, or tend to co-occur across the 24 soils we analyze, but the correlation isn't quite as strong, so we'll draw a thinner line. Now we continue and build a simple network diagram that describes the correlations between these taxa and their potential co-occurrence patterns. So again, this is just one way network analyses can be used in soil ecology, and this is one way it's commonly used, particularly when dealing with omics data. So I'll give you some examples of how we've used network analyses in studies conducted by my lab group. On the top left, we've used network analyses to explore relationships between bacteria associated with different moss species and measured rates of nitrogen fixation. This allowed us to generate new hypotheses about which moss-associated bacteria may be capable of nitrogen fixation. In the middle here, I'm showing an example of network analyses that we use to describe correlations between the abundances of various soil, bacterial, fungal, protistan, and metazoan taxa with different plant species. And we use these analyses to identify cosmopolitan taxa, that is, taxa that can associate with a wide variety of different plant species. And in the top right, one further example, we conducted a time series analyses of soil bacteria and fungi across seasons. And we were able to identify modules of taxa, in other words, groups of taxa that exhibit similar temporal patterns. And we were able to identify the environmental drivers of these taxon-specific temporal patterns, or these patterns exhibited by these modules of taxa. So I think network analyses can be useful for many ways, and this specific type of network I'm describing here is really useful for data reduction, reducing the complexity of the data to identify correlations or co-occurrence patterns, whether that's between taxa, genes, microbial processes, and even environmental conditions. And networks can also be used to generate testable hypotheses. Networks based on correlations, again, a very commonly used approach. These networks used in soil microbial ecology do not allow you to test those hypotheses or confirm their validity unless they're coupled with other evidence. So that brings us to an important caveat. There's always caveats. And networks are often, but not always, built from correlation patterns. And it's important to note that correlation does not mean interaction. And this is stated very clearly in two recent papers. Co-occurrence networks may be insufficient, I can't see that, insufficient in interpreting species interactions in microbiome studies. And another quote, even the simplest cases of interaction inference from correlations can fall apart. So when networks are based on correlations, we have to be very careful to recognize that those aren't necessarily interactions. Co-occurrence is not equivalent to interaction. And there's also a growing body of literature describing how network topology, or this overall structure of networks, may change over time with land use change or in response to disturbances. And network topology can clearly change with respect to overall modularity or connectivity or nestedness, as exemplified by these three example networks on the bottom here. But interpreting what these changes in overall network structure mean is often very difficult to do. We might be tempted to conclude that as one moves from left to right in these three networks, that the communities become more stable, but that's not necessarily true. And the assumption is often untested and based on concepts derived from systems, from very different systems and very different types of network analyses. So just to conclude, network analyses are no panacea. Network analyses won't magically reveal the mysteries of soil biodiversity. Rather, they represent another tool that can, in some cases, help us open up and help us explore that black box of soil biodiversity. Thank you. Oh, Marcel, you're on mute.
Thank you very much, Noah, sorry. I think it's very important what you said, that we will talk more about the functional implications of network structures and whether we can make any functional applications. So we'll discuss more about it. So the next speaker is Elly Morrien. She is assistant professor at the University of Amsterdam. And I actually know her already quite a long time, but I know her especially since a paper in 2017, a beautiful paper in Nature Communications, where they showed the progression and the intensification of interactions in below ground soil networks. I think Elly will talk more about it, so look forward to your talk.
Thank you for the introduction. Let's see if I can share the right screen. Can you all see the screen?
Perfect.
Yes.
Okay, perfect. Good. So welcome. So I personally, I started using networks during my postdoc position in a European project that aimed to link soil biodiversity to ecosystem functioning. This project was called EcoFinders. So maybe quite a few of you that are present know about this project. And yeah, we collected lots of below ground biodiversity data. And I was really struggling to get most information out of these counts of a wide variety of soil organism groups, microbes, but also nematodes, mites, and earthworms, and other groups as well. And so, yeah, we looked into a human disturbance gradient from abandoned agricultural soils to semi-natural grasslands. And via some good advice of colleagues, we started by simply correlating all individual members or OTUs of the food web to each other, resulting in a zillion of these kind of figures on this slide. So yeah, when that harvest actually is sort of a big hairball of interactions, we refined our thoughts and we set more strict thresholds and calculating overarching interaction strengths between functional groups in the way that also Noah Fierer just explained. So that was a very helpful introduction to our co-occurrence networks. So we found out that more natural ecosystems have tighter below-ground soil networks, so more connectivity, which co-occurs with more carbon and nutrients being in the soil food web loop as below-ground biota biomass. And the flows were actually measured, the carbon flows, in the same system, but in a separate experiment. And ever since I found this, I've been fascinated with two questions. So one is how carbon flows in food webs in natural versus human disturbed systems. So from agricultural systems to more natural systems. By tracing these flows within these 13C and microbes, and then looking into them in the guts of consumer levels, and then into guts of higher trophic level organisms to reveal actual links. And the second question is, can we tweak human influence systems such that we can store more carbon in stable carbon pools below ground and restore natural functioning of these ecosystems? And so far, my research has been mainly focused on grasslands. The major limitations of co-occurrence networks, in my view, is one, that it's correlated, was already Noah Fierer also highlighted this, so it might not reflect actual networks. And two is actually that soils are not a homogeneous bag of material. It has a clear 3D structure with soil particles and aggregates to determine how and if members of a soil food web might be able to interact or not. Problem is that this requires actually collaborations of soil scientists, physicists, and soil chemists and ecologists to resolve this. So in my opinion, combination of big scale versus detailed mechanistic studies are needed to reveal actual interactions below ground to overcome these limitations. So how can research on ecological networks advance understanding of response in below ground communities to global change? Well, I think the interaction strength between soil microbes and higher trophic level organisms in soils will become very crucial on many facets of global change in terms of soil temperature increases decomposition rates, but it also increases soil respiration and maybe carbon stock losses. But it also, in terms of prolonged drought, summer flooding, in the interaction between vegetation and microbes, and in the bigger picture on global carbon cycles. So the nature of these interactions will determine if global change effects will get enormously out of hand due to positive feedback loops, or if there will be some kind of way to turn these off. So thereby, I would like to give the floor back to Marcel.
Thank you very much, Elly. Exactly in time. Very good. The next, very nice presentation, of course, very intriguing results, lots to discuss. The next speaker is Alison Bennett. And I also know Alison already a long time. She's working on my favorite topic, mycorrhizal fungi. And she works at the Ohio State University in the US. And she will discuss now about networks. And she also worked a lot on interactions of mycorrhizal fungi, their effects on plants, on pollinators and herbivores, etc. Look forward to your talk, Alison.
Thank you so much, Marcel. It's a real honor to be invited to talk about a topic that I have been fascinated by for a long time, and I'm going to do it the old-fashioned way. I'm not going to use slides. And so I became fascinated by bipartite networks. So far, Elly and Noah have introduced you to unipartite networks. When we say uni, meaning one, that means there's one type of node. So you'll notice that in those networks, they're big circles, and each one of those nodes is this, each one of those organisms is considered the same type of node. But when I originally became interested in network analysis, I was inspired by the work of Bascompte and Jordano, who worked on plant pollinator interactions, and they used bipartite networks. And those are interactions, or those interaction networks in which you have two types of nodes. So from my great love, it was arbuscular and mycorrhizal fungi and plants. And I really tried for many, many years to try to get a paper published to using a network analysis of bipartite networks to understand the maintenance of the mutualism between arbuscular and mycorrhizal fungi and plants. But after 12 rejections, I gave up on that. And instead, I met Darren Evans fortuitously at a British Ecological Society event. And we had a data set that had been developed in partnership with Maria Opek and Tim Daniel, and he helped us analyze that using a network approach. And Darren and I continued to collaborate looking at network types of analyses using bipartite networks going forward. Today, more frequently in my lab, we use unipartite networks like the ones that Noah and Elly introduced to help us understand the differences between the composition of microbes that occur in different compartments between the soil and the plants. So that would be between plant roots, plant shoots, and the soil, for example. So what are the major limitations to using network analyses? And for me, I think because when I first started thinking about network analyses, people weren't, especially in the bipartite realm, people were not thinking about using soil. And I saw all these great advantages to soil systems. And actually, so instead of being disadvantages, I saw them as great advantages. One, we can sample temporally over time. So if you're at all familiar with plant pollinator networks, those take months to develop, and those all go into one data set. Whereas we could go out, if we had unlimited funds, and sample on a daily basis or a monthly basis or a seasonal basis and get that snapshot in time and look temporally through the network. And that's not really possible in a lot of the classical systems. And it also allows for significantly more spatially explicit sampling as well that you can't do in other systems. So I felt like soil is great in this sense. I would say that many of the current limitations that, in addition to the ones that have been mentioned by Noah and Elly, are also one of the things that it's difficult for us to do is to measure the intensity of the interaction or the exact presence of that interaction. Do we know if it's really occurring or it's not occurring? In plant insect interactions or insect-insect interactions, they're starting to do this, for example, by looking at the guts of the organism. And I expect to see those kinds of developments happening in this field over time. One of the things I've also noticed is that advances in analysis options are rapid. At least for me, they feel rapid because when I was first starting out in this world, we did not have all these different R packages that allowed us to do these types of network analyses. But I want to also emphasize, just as Noah did, that this is a tool, and most people kind of forget that. But I think it's a really powerful tool, but it's still a tool. How can ecological networks advance the responsible ground communities to global change? So for me, I feel like just watching, as I was mentioning just a minute ago, watching the changes and the development of new types of techniques for analyzing our data that can take things from being a correlative to being a little bit more predictive, not entirely predictive. But when we start to look at these new types of analyses like MAFA+, which allows us to pair network analyses with measured environmental or phenotypic variables, or comparing the composition of one unipartite network to another unipartite network that's under stress, we can do that using a package called NetCoMe. These are really new developments that allow us to answer questions that have been produced by our friends in statistics that really give us a lot more power to understand to links and what are the things that cause those links. We're still a long ways away. These are still correlative analyses, but there are big advances that have been happening in the field really recently. And I've been watching how quickly the field has been advancing new techniques from economics and physics. So many of you probably know that bipartite networks and unipartite networks were first developed as a field of study in really in social sciences and economics. And so seeing these new sort of movement of techniques that have been used in those fields over into ecology and evolution to understand soil systems has been very fascinating and happening at a much more rapid rate. So I would say that I still feel like we do not know enough about the assembly of microbial communities in soil, for lots of the reasons that Noah and Elly already identified, in order to be able to really apply soil networks to answer some of these big questions. I feel like what we're doing is we're producing gigantic correlations that we don't have a good meaning behind. And so as a result, I would say that argue that we don't really have enough knowledge to be able to make strong predictions for land use policy under climate change, for example. But I hope to see that change in the not so far future. Thanks.
Thank you, Alison, for your thoughts. And I think also about talking about assembly of networks, I think the next speaker, Tancredi, Caruso, will also talk about this, I think, a little bit. Tancredi is associate professor at the University of Dublin. He's a well-known soil ecologist, published quite a bit of papers all around themes around soil biology, and recently also had some nice concepts about networks. So I'm very much looking forward to your talk, Tancredi.
Thank you, Marcel. I'm going to share my screen. Here we go. Okay, so as the previous speakers have already highlighted, when it comes to applying ecological network theory, or better, more generally, network theory to ecological communities, and especially below ground ecological communities, we have a major observational challenge. Those networks are very difficult to observe. It is relatively straightforward to observe how species or different taxa co-occur together, but we don't really see those links that connect the nodes. And so we use kind of estimate those links either by some assumptions. For example, in the case of food web, if we find a fungal feeder, then we just drop a link because we know it's eating fungi. Or we use, as in the case of microbial communities, associations and co-occurrence data. So first fact is it's very difficult to establish where the interactions really are. There is also another element, which is the biological interpretation of nodes. That itself is very complex. In many application of network theories to object as complex as human beings in a social network or computers in a computer network, there is a clear interpretation of what the node is. In classical ecological network, plant and pollinators, that interpretation is that a node is, for example, a species. But when it comes to microbes, all those assumptions become much more complicated. If you assemble a network of OTUs or ASUs, the implication, the biological implication, then become very, very difficult to work with. And so that's another challenge we have. And also weighted networks, so what networks where we know not only where the links are, but the weight of the links, those are also difficult to estimate, especially when it comes to things like the strength of interactions between, for example, AMF and plants. These challenges are essential in terms of the interpretation of the patterns we observe when we describe networks, because they are essential to answer questions like, do networks rewire over time? Do networks rewire in response to perturbations? Do interaction strengths redistribute over time? Do network changes affect ecosystem level changes? And the answer to all those questions really depend on those challenges I just said. And but what I'm going to say here is that it's not all lost, maybe, because despite those challenges, we do have something to start with. And there are techniques used in network theories that can be used to basically use data like correlations and associations to estimate links, either where the links are and their intensity. Starting from known observed node level properties, for example, the degree, which is the number of connection to any given node, we can enforce constraint and construct ensemble of networks compatible with all the available information. Then, of course, it's a question of the quality of the information, but given the available information, we can constrain networks in a way that all possible topologies, even those that do not exist, but are potentially compatible with the knowledge we have, we can construct them. And this is called, there is a technique which is a statistical mechanics of networks that allows actually do that kind of job. And why is that useful? It is useful because it will allow us, it actually allows us constructing robust null models. Those are different kinds of null models than those normally used in ecology, but you can think of them as a randomized ensemble of networks that are compatible with available knowledge. And they are being used a lot for the analysis of economic networks, for example, or financial networks, whenever the information available on the network was not very much. In fact, in some cases, we actually have more information for ecological networks than for financial networks. And so the thing is that once you have the ensemble and you have a good null model, you can calculate any network metric on the null model ensemble as well as on the observed network, whatever that means, whatever observed means. And then you can start searching for patterns in a way that is based on the reconstruction of the network. And this techniques can be applied, for example, to plant microbial networks and also to food webs. And so part of my research at the moment is actually exploring those techniques and the application of those techniques. And why is that useful? Well, I think an answer can come, for example, from applications of those methods to financial networks. If we, you see here the citations there, if some network properties had been monitored using these tools, early warning signals of the 2008 financial crisis, for example, could have been detected already in 2005. So, especially if you have a minimal time series, and of course the dream will be having it for social community networks, could in theory apply those networks to those network techniques to detect early signals, early warning signals of how social community networks respond to perturbation. That would be amazing if we could do that. Here you see an example from financial networks. Yes, I would say that with that, I will just give it back to Marcel.
Thank you very much, Tancredi, for your thoughts and very interesting thoughts. So I see that there are already questions dropping in from various people. So everyone who has questions, please answer them. I think the first two questions were from Johan Ekdal and Nicolas Montoya, to various of us. So they ask for packages and good text, etc. So one suggestion is, I think for the YouTube video, we could propose some suggestions. Please, speakers, panelists, give your thoughts on this.
So I can definitely drop the two papers from the analyses that I mentioned into whatever format works for people. I'm not sure what that would be best, but I can definitely send the links to at least Marcel or GSBI, and they can post them with the YouTube video.
I'm happy. I saw a question about statistical tools and network analysis, and given, yes, something I kind of work on. If you're happy with that, I could kind of give a general answer to that question. And I think the answer is there's just an embarrass of choice, really. There wouldn't be, there is no one single method. I would say actually the application of the method is very much question driven. And this is exactly because of the observational challenges we have with those networks. And I would even say that even when you have one question, even the way at which you aggregate the data, for example, especially if you have molecular data, even the way you aggregate and you construct your molecular data level, what may determine the tools you will use to analyze the networks. There are two possibly, and promise I would just one more minute on this and then I shut up. I would say there are two main ways. One is the most dominant way in ecology, and that is basically calculating a metric, I don't know, clustering coefficient or nestedness, and then having some form of null model, which is based on randomizing the data and test for the statistical significance of that metric. And there are many ways of doing that. And in fact, there is lots of debate around which method would be best. And there's another approach, which is much less known to the ecologists because it's been used mostly in things like the analysis of computer network and financial networks, and that is reconstructing the network given little pieces of information we have on the network. For example, in the case of financial networks, we never know which bank or which financial institution is interacting with each other because there's a privacy law there. So we don't have access to that information. And yet, some of the most successful applications of network theory are on those kind of systems. And then in that case, the approach is different. It's not really searching for patterns on the basis of a metric and a model, but it's trying to reconstruct a realistic model of the network and then search for patterns.
I think that's a very nice thought. Noah or Elly, do you still have some suggestions? I think we also should just provide a list with some textbooks, some papers, some packages. I think that will be very helpful. But do you have some further comments, Elly or Noah?
Well, if you're not like super handy with R or so, and in programming, there's also a nice program which is freely available on the web, which is called Cytoscape. Well, you do have to create a co-correlation matrix first in any program that works, and then you can import that with nodes and edges, and then you can play around in Cytoscape with different configurations of creating a co-occurrence network, and you can set thresholds, play around with it. But of course, it's very important, as Tancredi mentioned, to just be aware that this is just a assembly and that it might not always reflect an actual pattern that's going on. So it's very good to also create a complete random network of your same nodes, with just some random data and just see whether that does something different than your created network that you imported.
Noah?
Yeah, I think there's, I think there's really two decisions when you're building networks, two broad categories of decisions. One is how are you actually going to put the lines between taxa or between taxa and environmental conditions, or taxa and processes. So how are you going to identify those correlations or those vertices, as they're called in network terminology? So that's sort of one category of questions to figure out. And once there's a whole wide range of tools to do that. Like, for example, you can even build in a time lag there between different taxa, for example. And then the other question is, how do you actually visualize, right? You end up with these hairball plots. So how do you actually visualize these plots in a way that's interpretable? And then as Tancredi mentioned, there's also a lot of strategies for how do you describe the overall structure of the network and show that it's more structured than it would just be by random chance. So there's, as Tancredi said, it really depends on your question, how you want to build these networks. And I think those differences are really important to keep in mind when you're using these tools.
Yeah, I think a lot of good suggestions have already come up. What I personally also like is sometimes to use different programs to tackle the same question and see if you get the same answer, because sometimes the answer might be very much dependent on the statistical tool you use. And I think that's also very important to really find robust conclusions. Yeah, I think I propose we move on with some questions. So we will put them on the YouTube at the end, some suggestions. There is a question from Ben Sykes about these correlations, as if he suggests, and I think that's a very good thought, that they are like catalogs of species. So it's very difficult to really think what it means, what has already been discussed. Are these correlations really doing something, or is it just by accident correlation because two species respond in the same way to pH or soil texture or whatever? So maybe a question to the panelists, how to solve that? I think that's the big question for many of us. How do we get around it? Can we do experiments to test such things? How do you see that?
Sorry, maybe I'll jump in here. Sorry. I think two things. One is there are examples in soil ecology of networks that actually describe interactions, right? For example, this mycorrhizal fungus was found on these particular plant species, or tracking carbon flow from plants into different fungal taxa. Second, even if they're not direct interactions, so again, correlation isn't necessarily interaction, it still is to some degree describing co-occurrence, and that can be useful. So for example, if we're looking across gradients in temperature or soils that represent gradients in temperature, we can say, okay, these taxa are always appear to be sort of correlated with temperature. And again, that's where the data production comes. Or here's taxa that tend to associate with this ecosystem process. And maybe we don't know what microbes are responsible for that process. So the networks might allow us to generate some hypotheses. So I think we just have to be careful to say if it's strictly correlation based, it's not necessarily saying there's interactions, but even knowing a co-occurrence or potential co-occurrence can be really useful.
Yeah, maybe I have also an addition there is, I think what is also good about it is, for instance, 1,000 taxa, some of them correlate with each other, but many do not correlate. So you can filter out a lot of potential, relatively unimportant interactions. So that's, I think, also already a strength. You can filter a lot out. But I think, Elly, you are nicking, so I think you have many things to say.
Oh, well, I fully agree with what's being said before. But it's absolutely true that especially if you try to correlate very much different groups. So for example, microbes with mesofauna or so. And of course, mesofauna have just a smaller amount of different species that can be present, or might have like few species present, but then in very high numbers. And especially if you then combine that with your microbe data, it tends to trump your correlation. So if you find really strong correlations there between certain groups, that might just be because of the data that you're entered. So it's very important to be very cautious there. But as Noah said, it's very helpful tool just to generate some hypotheses for further experiments, to actually look into if these links are actually functional in terms of flows of nutrients or carbon, or if these are actually happening because you can trace the carbon atoms, for example, that end up in a certain group, and so that you can establish your feeding relationship just by measuring them.
Alison, do you have something to comment?
I would say that, a good third of ecology is looking at correlations. And so we've learned a lot from that. So yes, absolutely, we're looking at a lot of correlations, but that doesn't mean that we can't learn anything from it.
Tancredi, do you have also some comments?
That we can also do experiments sometimes, and this is very true for example, plant microbial interactions. And these, if the experiment is well planned and there's good modeling behind it, maybe some of those correlations can make a bit more sense in terms of, for example, establishing whether a correlation is a correlation or an actual interaction. Surely those are very difficult experiments to do. And one element of difficulty there is that, at least in terms of what we have learned, for example, from plant pollinators networks, that are networks we know well, because we actually see the thing going to the plant. So we actually see the bumblebee getting to the flower, or the birds. So in that case, we know, and we actually know even something more. We know that sometimes co-occurrence is not interactions, because we actually can observe a visit, but differentiate that from a natural pollination event. Okay, and so this is where maybe some experiments in the field of soil ecology could be very useful. They will be difficult to perform just because just by nature, networks tend to reconfigure, they fluctuate. And also an interaction that maybe is important in a particular location is not important in another location. And when it comes to microbes, this is particularly true if you think that the genomes at the microbial level entire are harmonized with the local population of plants in the particular location. And so surely those experiments are challenging. But I think really the way forward is the experiments. We need more experiments, possibly field experiment, because this is the only way to make our correlations a bit more substantial in terms of whether our network model is really describing something or is just one more way of looking at correlations.
Yes, Tancredi, I'm not sure if it was you or Noah who presented this, but also just measuring these networks at different time points during the year or over months it gives you also a good idea which links are really robust and which are maybe more random or occasional. So I think I go to the next question by Hannes Schmidt. It's an important one, a very important one, very actual. He writes, to better understand aspects of the soil food web, it would be important to combine data on the different group of actors, such as bacteria, fungi, protists, viruses, etc. However, sequencing data underlying interkingdom networks are even more affected by technical challenges, just the primers we use, for instance, and biases in nucleic acid extraction. So are there opinions on how interkingdom interactions could be done, standardized to really give a good representation of the soil networks and for good hypothesis for soil research? So very good question. So about the importance of interkingdom interaction. And I think also what we see in some people focus on bacteria, some on fungi, some on protists, and often these are kind of different kind of leagues of people, different Champions Leagues, so to say. And it's coming now, the interaction, I think, between different groups, but it's as Hannes Schmidt says, it's technical challenging because you need the expertise of these primers. So please give your thoughts about it. Elly, do you have something?
It's not so simple to answer.
Yeah.
I fully agree with that. So I think we should not always, and that's a bit scary to say this at this time, but only focus on these molecular techniques, because I mean it all comes with these technical challenges of if we have the right primers and if we pick up the right species, and if the PCR bias is not such that we are actually not looking at the most abundant ones, but just the ones that are picked up just by methods. So yeah, of course, there are movements now more into like methods that are separate from this PCR bias, but also I think a combination of the new methods with sort of old-fashioned methods, so to say, with plating, making small mesocosms to study specific interactions that seem very interesting in your network are super helpful in actually revealing the actual links in these interkingdom relations.
I would second that, Elly. I think we are certainly limited by culturing capability in soil. However, these new approaches using the sort of mock communities and the mock mesocosms are really starting to make some dramatic advances that I think are really helping us to understand what's happening in the broader microbial community.
Just to add, I would say, all methods are biased, full stop, right? The question is, how important are those biases for the questions you're trying to ask? And I would say, I think one great application of network analyses is actually to look at these interkingdom or intertaxon correlations or co-occurrence patterns. And an example of that is viruses and bacteria, viruses and fungi. Viruses, we know they're common in soil, really diverse, and that's kind of all we know, right? So one way people have been using network analysis is to say, okay, every time we see this virus, we also see this bacteria or this fungus. And so now we have a hypothesis. Okay, maybe this virus or this phage is specific for this bacteria or this fungus or this plant species, what have you. So then it gives us some hypotheses. Instead of just being overwhelmed with the complexity, we can start narrowing it down and come up with some specific hypotheses we can use. And network analyses are particularly good for that because very rarely is a single virus associated with a single bacterial species or strain or what have you. So we can start seeing, okay, which viruses might be capable of infecting multiple different taxa versus those that are more restricted in their ability to infect.
What I'd like to add is that just when you were saying that question, when you were reading that question, Marcel, the first thing that came to my mind was the nature of the nodes, because networks are models, aren't they? So, and part of the modeling is what we decide to put in the nodes. So in this case, it was a very high taxonomic level, actually, interkingdoms or domains. But that's a very fundamental question. And I have a gut feeling that in ecology, we tend to underestimate that question very much, especially when it comes to microbes. Because if you apply network theory to a set of computers in a network, you know what the node is. It's a computer. That's it. And you know what that brings in. But when it comes to biology, you have to make decisions about what is the most appropriate level of biological organization at which you define the nodes. And that will cascade dramatically in terms of everything you see afterwards. So this should be probably an important reflection there. This is, for example, when it comes to food webs, this is a very important topic. Trophic species, should we just work by species, even if they are redundant in terms of trophic functions, so we just lump everything together, but then we have so many different things there. It comes down to how our model network matches with the reality of the object we actually modeling. It's a very philosophical thing, actually. So the actual mapping correspondence between the model and the actual nature of the objects we are trying to model. That is fascinating. It's very fascinating. So the negative side of it is that if you just get that wrong, everything you say could just be air, because you're speaking just of your way of aggregating the data. But if you nail that round, you're actually revealing patterns in nature. As Noah also said that you have a pattern and you have an hypothesis about, for example, the association between particular groups of viruses and bacteria. And that's working because maybe the data in that case were aggregated the right way at the node level.
Okay, yeah, I'm moving on. There are many questions. Also, I would like to mention what Noah said about viruses. I think now the first papers, or at least I've seen the first papers, maybe they're already out for a longer time, but it's getting mainstream slowly. I think this is really a cool research area because it seems they are so extremely important. When I studied biology, that's a few years ago, then we didn't really think about viruses in soil. So we thought about viruses, of course, but not that they also affect soil processes and soil networks. So I think it's very interesting. I would like to move on. Again, a question for Lakshmi Gopakumar about statistical tools. I think this will be addressed. Then another question by Nicolas Montoya Royas. How might a business focus on soil health and regenerative agriculture, regenerative practices in agriculture in Colombia, using network and articulate with your research groups? How can we use also soil networks to tackle these things about soil health and regenerative agriculture? Do you think that soil health is higher if we have well-developed soil networks, or are these soil networks unimportant for agricultural production? What are you thinking about this? Alison, you are laughing, so you have, I think, some thoughts.
Yeah, I mean, so there are a couple of different ways that you could look at this. I mean, I think soil health is the buzzword in sustainable agriculture right now. And certainly there has been a fair amount of work trying to address this question in a lot of different ways. And networks certainly have been applied to answer that question. I think one of the mistakes that people make is the assumption, I can't remember if it was Noah or Tancredi who put up a set of networks showing that the different numbers of edges. So you had a really connected network and a kind of connected network and a poorly connected network. And there's an assumption that this poorly connected network is going to lead to something like lower soil health. That's not actually known to be true. And so making that kind of assumption, lots of people go out and say, oh, I know I've got a healthier soil because I've got a more connected network. And at the end of the day, I think the movement towards trying to understand what the functions of the organisms in the soil and what role they're playing in soil health is going to become a real key for understanding whether or not your connected or non-connected network is any indicator of soil health. So I think we're making baby steps in that direction to understand that, but I don't think that we can say, you know, if you go out and you build a network in your soil, if you're, you know, what you think to be healthy soil, what you think to be poor soil, conventional tillage, you know, all of that sort of thing, it's not necessarily going to give you, the co-occurrence network is not necessarily going to give you all the information that you need to answer that question, I would say. It's going to get you along that path, but it's not going to give you all the information that you need.
Yeah, I think Elly, you worked a lot on this with your earlier, what you also showed. Maybe you can give some thoughts because your data indicate the more connected, the better the soil health, so to say, but maybe it's not productive anymore.
Well, I mean, that was a gradient of agricultural fields that were already taken out of production. And so my hunch is there indeed that if you feed your agricultural soil with lots of fertilizers and you have like specific practices such as tillage or pesticides amendments, that these sort of also make that microbes are, well, of course, some microbes simply cannot live there anymore because if you plow through high-fall threads, then so that's also why these systems are often also much more bacterial dominated. And also if you provide lots of nutrients, then also I think in your network, your below ground community is less dependent on each other in a way. And that will probably also show. So you have probably more simple networks and more, less connected networks. And of course that relates, well, it might relate somehow to soil health, but it's not one-to-one. And it's also not, it's also not the case that we need agriculture to feed ourselves. So it's also not a realistic idea that all networks should be in the same way or as natural networks or communities are. So I think it might give some indications, but I think maybe networks are, maybe this is a scary thing to say, but maybe networks are not the best way to assess soil health in general. Because it's not only the biodiversity and the biomass that is there, it's just mostly the functions that they perform in your soil.
That's an interesting hypothesis. I think I have a question maybe for Tancredi. So it's also about this regenerative agriculture. So what Elly said, that these networks are more simple in these highly disturbed systems. So would it be, it's a bit a different question, but would it be interesting to add particular network components like mycorrhizal fungi or trichoderma or some bacteria which can kind of, that you boost the network complexity to improve soil health? What do you think of this? It's an open question based on this question we just got.
The relationship between the stability of a network and its complexity is a long-standing question in ecology. We know it very well in terms of soil food web complexity. We don't want to forget soil food webs are networks, but it's generally true. Like it's true in general, and it comes down to the general question of what happens if you have a shock in one part of a network. How does it propagate? And the way it propagates, does it depend on, for example, degree of connectivity? Or is it more dependent on intrinsic attributes of the nodes or the intensity of the node. So it could be maybe dependent or partially independent from the topology, so who is connected to whom. But it could be maybe that's the distribution of the weights, so basically how thick the links are, that could determine the way the shock propagates. And there are no general answers, as always, to this kind of questions. If you analyze general networks with lots of randomness in each, complexity actually works against stability. But of course, natural communities are not random at all. They have very important structures. And so there are aspects of complexity of the networks that seems to make the system more robust. And the state of the art for below ground ecological network, I think, is such that we don't still know the answer to that question. Although for particular types of networks, for example, soil food webs, there are theories and there is some evidence about what are the aspects of the soil food web structure that could give stability to that type of networks. Mostly hypothesis with some evidence behind it, a number of experiments still running, I would say, to try to test those hypotheses. So that is a very difficult question, to be honest. It is difficult at theoretical level, at empirical level, and in this case, those kind of challenges we have when we try to describe soil, below ground soil communities, they become particularly problematic because, they don't really allow us to answer that question in a general manner. There are answers that are system dependent, for sure. And then it will go into specific pieces of literature.
I think there will be companies who will pay you a lot if you can, which components of network will make an agriculture productive and stable and so on. So it's a good point. And Noah, do you have?
Well, there's actually one thing, and that is if you increase the amount of organic matter, your network complexity is also going to significantly increase. And that is of course also a high relation with soil health. So that's a very, very simple way.
Just to add my 2 cents, I think it's a great question, right? So how do we move beyond just physical and chemical analyses to determine soil quality and incorporate information on microbes or other biota in soil to sort of provide a more integrated and comprehensive view of soil health? Because we know microbes and other organisms in soil do all sorts of important things. So it's an obvious next step to say, okay, let's incorporate some of this information. And I'll give 2 examples of how networks could be used. So I don't think we're quite there yet, but one is, there's a lot of literature on microbes in soil and disease suppression. So we could use network analyses to at least identify, okay, these taxa tend to be associated with the abundances of these particular, for example, plant pathogens. And now we have a hypothesis that we can test. Okay, why might there be this association between these particular taxa and the abundances of a particular plant pathogen, for example? Another example, we know that there's a large body of literature showing that phosphorus availability is really important. Phosphorus availability in soil is really hard to measure. So, we can use network analyses to identify microbial taxa or even microbial genes that are associated with phosphorus availability and essentially use the microbes to tell us how much phosphorus might be available in soil. And so this isn't a new metric of soil health. We've long known that phosphorus availability is really important. But, these microbial analyses are getting cheap enough and high enough throughput, it's relatively fast to do these analyses. We can start using microbes coupled with network analyses to infer some of these soil variables that we already know to be important to help guide management decisions in agricultural or even unmanaged systems.
Yeah, I think these were very good points from all of you. I think also there are actually companies working on that, and I think there's a lot to do here also to make these links indeed. So go beyond the description of networks, and as you say, these biological indicators. I think also policymakers are very interested in indicators of maybe soil nutrient availability, but also of pollution or pesticides, stuff like that. So I think it's a very, very important point. I would like to go to a next question. It's about keystone taxa. Maybe Noah can answer this, but I think also Elly and Tancredi is what is the best way to identify keystone species in soil samples using network analysis. We also worked on this. Noah, you can, I think.
Yeah, so the whole idea of keystone taxa actually emerged from network analyses, primarily from food web analyses. And it's the idea that there's particular taxa that sort of have outsized interactions or influences on other taxa. I'll admit I'm a bit skeptical of the idea of keystone taxa, and there's a lot of literature on this. And identifying, again, for reasons that have already been discussed, just because certain taxa tend to be correlated with many other taxa does not necessarily mean they're interacting. And I would argue that keystone taxa, you have to show some degree of interaction. And there are ways to get to that. So I think it's just another way to, essentially keystone taxa is a way to sort of summarize networks and say, all right, here's particular taxa that are well connected with other taxa. But as with all things, it has to be taken with a grain of salt.
I fully agree. Maybe I can also comment on that. I see keystone, I see it relatively pragmatic. We have these 1,000 taxa, million, or not thousands of taxa in the soil, and we need tools to kind of identify important components. And so one way is the connectivity of networks. One way is also these keystone taxa. And then, as I think Tancredi and Elly also said it before, what is very necessary then is to perform experiments, maybe just manipulation experiments at these keystone taxa to communities and see indeed if there is a response in decomposition or nutrient uptake. And I think then there is true evidence. But indeed, it's of course correlation, but it's one of these nice tools which you can use to characterize these very complex communities in the soil. So that's how I see it. So I see it pragmatic, and I think it's useful. Tancredi or Elly, do you have further suggestions, comments on use of keystone taxa?
Yeah, I think there are two ways to look at them. So if you create a network and you find these hubs of nodes that seem to interact with a lot of species, then that might be a good starting point to find out what these species in your network are actually doing. And the other way around could be that if you have like some very key functions in your system, that you find out which species seem to relate most to these functions. And if that sort of comes together in the same sort of species groups, then I guess you could say that these might be important. But yeah, it's also, I mean, it's also a little bit of a just a term, because I think there are a lot of very crucial groups of microbes and higher trophic level species in a soil that we might not always call keystone species, but yeah, play very important roles. But I mean, you have to start somewhere, and that's what I agree with Marcel.
Tancredi?
I would add that maybe, yes, we can also broaden maybe the definition of keystone species in the case of soil, because typical idea was from food web, actually, I think. If you pull out a major player, a predator, typically, the system collapses. But in soil communities, it's difficult to pull out something and then something dramatic happens. So many other things there that interact. It is, however, maybe more easy to bring in something that could bring in disruption. For example, a pathogen that causes an outbreak in the plant community, especially if it is a production system. Those type of organisms could also be considered as species or node network that have a disproportionate effect on the structure of the community than the others normally have. And so maybe from that point of view also, yes, that concept could be very useful as well. Maybe especially for when it comes to connecting the below ground network to the above ground, so plant communities versus soil communities, because of how we describe the diversity of plants and what we know about the biology of plants, it is possible to identify some plant species that play a sort of role, this kind of keystone role in terms of shaping the soil community in a way that it is analogous to what a classic keystone species could do in a food web.
Thank you. Yeah, I'm moving on. I'm afraid we will not be able to answer all the questions which have been posed. I see very many interesting questions. Here, I think it's also an interesting question from Marnix van de Zande. could one of the speakers compare the use of network analysis to the use of other multivariate statistical methods like principal component analysis? Where are the differences in use and cases for soil biology? So compare network analysis with multivariate statistical methods like principal component analysis.
Okay. I would say that we kind of already touched on this in the sense that network analysis used in a sort of, say, pattern description way is fundamentally very analogous to multivariate statistics in the sense that you use a large correlation matrix to explore multivariate patterns of correlations in your data. So in a way, they do the same job, but in a different way, and they visualize the data in a different way. Where the difference could really be is when those pattern of correlations make, bring some pattern that can inform on underlying mechanisms. For example, how it assembles or how it rewires in response to perturbations. And this is where all those challenges began. So in fact, it's the way we apply network theory and we assemble the data, which can add something to our ordinary weaponry in terms of multivariate statistics. If we do not resolve all the things we have discussed today, our way of using network analysis remains very similar to a different kind of multivariate analysis. At least that is my opinion.
Yeah, there's another question, I think, and this has not really been addressed, and I think it's very interesting, is from Adrian Unk. To what extent are nonlinear co-occurrence relationships considered in these networks? That's a good question, I think. Is someone doing this?
I could do this, but I feel like I'm speaking too much. So if one of the other two maybe wish to tackle this.
Yeah, I think it's a good question. It gets to that, the first step when you're conducting network analyses is how do you put those lines between taxa, between taxa and genes, between taxa and processes, and so on and so forth. And one way to do that is simple, like linear correlation, but that obviously will miss some of the dynamics that could be important. So there's all sorts of complicated ways, more complicated ways people have developed to look at that. You can do simple rank correlations. You can try to fit more complicated nonlinear relationships, identify more complicated nonlinear relationships between your nodes in this case. So again, it comes down to, I think it was Tancredi that said earlier, it really comes down to the questions you want to ask. And it's difficult, right? I mean, there's a lot of complexity in soil, both in terms of number of nodes and the types of potential interactions that may be occurring. So there's no simple answer to that, I'd say, but there's a lot of strategies, and there's a very good, lots of really good reviews on sort of the mathematics behind network analyses. So I'm afraid I didn't really answer the question, so I apologize.
I think you very clearly touched about issues. I think it's also about choices you make when you do these analyses. I would like to go to a next question, and actually I fully agree. It's from Stefan Geisen. He says networks and correlations are always proposed as hypothesis generators that suggest experiments, but who does those experiments, as they're usually much more work than simply sequencing. I also feel it's more rewarding to take some samples and sequence, and you can make fantastic graphs of networks, simple, more complex networks, but to do the experiments, to manipulate thousands of taxa, it's basically impossible. So how do you see that? I think it's a good point, and I also feel a bit like that. Elly, maybe?
Yeah, that's a very good question. So I tend to do lots of crazy experiments, actually, really trying to reveal actual interactions. But yeah, they're a pain. I'm not sure how to overcome that, actually. So I do think that we really need to keep combining these very fine-scaled mechanistic experiments that are actually revealing what is going on a very small scale in the soil. Because yeah, all these species are not just they cannot just all interact with each other, but yeah, one might be in an aggregate while the other one might be attached to a root, and they might just never meet in real life. So I think we need to be aware of these things, and yeah, just with each sort of the mechanism that we want to reveal, just think about the scale where these interactions might operate or not, and then think about the smartest way to design your experiment, given that scale, to make sure that we don't make large mistakes there.
I agree 100% with Stefan's comment. It's very easy to generate hypotheses. It's much, much harder to test hypotheses. But I think that's critical. And to use an example, like let's say we say, okay, we do a bunch of sequencing. We say, all right, these taxa tend to be associated with disease suppression in soil. Great, that's a hypothesis. But to really make that applicable in field situations and actually use that information to improve crop productivity, for example, requires testing that hypothesis. Which specific taxa might be influencing the growth of known plant pathogens, and how, and until we know that, we have, if we want to, for example, add particular microbes into soil or onto seeds to limit disease or limit the growth of the pathogen, we need to do that hypothesis testing. And it's difficult for many reasons. So we'll be in business for a long time because none of these things are easy to do.
That's a very good point as well. Yes. Tancredi, do you still have some points there?
Yes, that there are different kinds of mechanistic experiments, and you do not necessarily need to manipulate every node or every link. So some experiments can be a bit more holistic, so to speak, where you have entire communities, but they are responding to something you are imposing. And that's the difference. And it may remain speculative in some instance, but it's been you driving the system towards certain patterns or others. And that makes a big difference. It is an experiment.
I'm going to another talk. Also, I like this one from Huswan Wien Chen. Hello, nice talks. My question is, what would be the proper unit to be analyzed in multiple cross-taxa networks? And also, what do you use? A genotype, a species? What is a taxa? What is a real unit? And how can you use that in building these interaction networks? Very essential point as well. I'm sure Elly, Noah, Tancredi, you have thought about this and done this also. Just defining a taxa, a genotype, or a different sequence. What is the level? What's the unit?
Yeah, I think Tancredi addressed this earlier, just saying, how do you identify those nodes is really important. And I just want to sort of maybe not answer the question, but highlight that the nodes don't necessarily have to just be taxa, right? They could be genes, they could be metabolites, they could be particular environmental conditions. But I think that's an important question to address even before you start a study, is how are you going to identify these nodes? And yeah, I mean, even identifying genes, okay, well, what % similarity do those sequences have to call them the same gene? And that's a really important question, and it can lead to all sorts of different results if you use different definitions for the nodes. Yeah,
I fully agree, and I think the right unit just depends on the type of question that you ask. So if that needs to be answered on a gene level, or if that needs to be answered on a functional group level, that just depends on your question. But it's definitely important. And yeah, when I made the network in the paper from 2017, I later also just thought, because yeah, you include microbes, which are identified on a much different level than, for example, the community of mites or colambula that I included. And for sure that does have an effect on your network. So it is I think it's very important to compare, or to have preferably the same unit in your network to avoid wrong conclusions because of the way that you construct your network.
Yeah, there's another question which I think is also quite a nice one. It's from Francisco Teles Varela. What about the state of the art around quorum sensing and how bacteria, for instance, start to function if a certain quorum, a certain minimal amount is reached. So how do you see that to be incorporated in networks? And can this also be used? Can this be used then also for plant and soil health? Even more complicated question. If the scoring sensum of particular taxa can be related to soil health, and if this also can be catched with soil networks, or is it a very different question at all? Should we not touch on this because it's getting too complicated, too non-linear? Maybe it's not possible to answer this.
It's definitely complicated. I mean, I guess I would say, you know, one way you could use network analyses with regards to quorum sensing is, as I already said, you know, the nodes don't have to be taxa. They could be metabolites, or they could be known quorum sensing molecules, or even unknown quorum sensing molecules that we think might play a role, but we're not sure. So you could start looking at correlations or co-occurrence between particular molecules, even known or unknown quorum sensing molecules, and the abundances of particular taxa, again, to generate hypotheses about how these quorum sensing molecules might be important in soil systems.
Do you know if someone is using this? For me, this is a bit new, so I'm really wondering if some of the panelists know if people are addressing this.
Well, I mean, I'm not fully aware, but yeah, I guess it actually asks for experiments that are done within very short time spans, so that you can see the sort of waves of signaling molecules that are causing a reaction with adjacent groups of microbes. And so, I mean, I know that there is now quite some people that work more on the chemical communication between microbes, looking into volatiles, networks of volatiles. So there's definitely information about this, or research done about this, or in progress about this. And I think this is a very new and exciting field.
Yeah, we're slowly actually approaching to the end. I think in 10 minutes we will actually stop. So there are still various unanswered questions. So if there are very specific questions for some of the speakers, of the three speakers, so please give them here. Otherwise we can just move on with the questions. Another question here is, which comes back from various people. So Alejandro Berlinges, how could we add functional data of organismal groups to create interaction networks in soils? I think this has been partly been addressed. We talked about genes. Can maybe Tancredi give some thoughts on this? So functional data, I think this is the big question for all of us working on soil networks to make it functional in the end. So could you give your thoughts, or maybe what experiments would you propose for the future to tackle this?
Yeah. So. Yeah, this is a very vital question, isn't it? Because the structure of the networks should be driving, I mean, ideally, you want a structure that is connected to a function. That, you know, and many of the things we've discussed are in terms of, well, we are in a state of the art where we're mostly describing patterns. We don't know if those patterns are really reflecting how structure affect functions. And this is exactly where that kind of data, the gene level, the functional gene level, may actually fill the gap. And the key point there is what Noah says, which is the nodes don't need to be taxa. They can be other things. They can be genes express functions, and the links can be expressed in terms of the rate at which a particular function is happening and is connected to another particular function. So the answer is definitely yes in terms of the applicability of the methodology to that type of data. And in fact, we should really be motivated in doing that, because this is exactly where we'll fill the gap between the structure of the object, which is our model of the network, and how the system functions. In food webs, this is partially done. There have been attempts, because in food web, the key currency of the links is energy flow. And of course, energy flow is connected to respiration, is connected to how carbon and nitrogen move through the food web. And so if you nail down the structure of the food web correct and you can experimentally measure it, then you actually can estimate in principle fluxes of carbon and nitrogen, right? And so this is what we would like to do at a more general level when we, for example, assemble plant microbial networks or functional gene level networks. So it is something which is totally possible to do, and network methods do allow doing that. With the caveat that we need to solve a few problems as we've discussed for an hour and more so far.
Yeah, and to be honest, but this might be a bit of a scary saying this in this community, but I think well, at least co-occurrence networks might not always be the right tool to pick up these things, because there are so many different functions going on at the same time in a soil, and then all imposing effect on network structure. So then it might be hard to pick up this one relation between this one function and the structure of your network. So I'm not completely sure whether that's always possible to overcome this, although of course there are very particular paramount functions that are going on a certain moment, which can then be related to that structure of the network at that moment. But if it's especially if it's measurements that are not being taken in time and difference in networks in time, then I think it's very hard to relate it one-to-one to specific functions.
Yeah, maybe also comment to Noah is, I think you're very familiar with it, that the whole sequencing power is increasing and increasing, and so now we can also do transcriptomics and metabolomics and link such things to networks. So how do you view that for the future? Where do we go? Because we will get
Yeah, it's a great question, because we're getting pretty good at saying, all right, here's the types of taxa we find in the soil, right? But it's clear the next step is like, okay, many of these taxa, we don't know anything about them. So how are they surviving in soil? What are they doing in soil? What processes might they be contributing to? And so I think that's one place networks can be useful, is more for the data reduction aspect. So for example, let's say we see a module or a group of taxa that are consistently associated with changes in nutrient availability. Okay, now we can say, okay, what genes or metabolites might those taxa have in common that are not found in modules that respond differently to nutrient availability? So we can start using this to, again, generate hypotheses about the traits, or at least genomic traits or potential functional capabilities of those microbes that respond in a similar way in soil. Again, it's not the be all and end all. It's just another tool that can be used to start mining the complexity that we find in soil. I mean, that's the hard part. I mean, that's the beauty of soil, is the complexity, but it also makes our jobs difficult.
Yep. Here a very different question. Naomi Zurcher: as an urban forester, I deal with trees and their soil-associated deprivation within the building environments. Is there any research on urban soil networks, and what is missing to assist with restoration strategy? Do we need to consider microbes there? I think also what I mentioned earlier, maybe adding microbes that are missing in very intensive environments. So do we have some? I think we have still 1 minute afterwards. I think I have to sum up, then we're slowly stopping. So someone answer to the urban environments and networks.
Well, honestly, I don't have lots of experience in urban situations, but I've been playing around a little bit with trying to steer microbial communities, and it actually works best if your community is really disturbed, or there's just a lot of empty niche spaces in your system. So if you then introduce something that might be very helpful, yeah, that could really work actually. So it's that's not a crazy idea at all.
Okay, yeah, I think we slowly have to stop here. So I would like to thank Elly, Noah, Tancredi, and Alison, who had to leave a bit earlier, for their very nice talks. Very enjoyed it. And also, of course, the Global Soil Biodiversity Initiative, Diana and Monica, for organizing this wonderful webinar. So I think we stop in 2 minutes. So it gives me maybe a few minutes to summarize what we discussed. I think what we discussed, and I think all speakers agree, these networks are wonderful tools to generate networks, to generate hypotheses, and to also simplify structures below ground, because it's so extremely complex in the below ground. So it's just a very easy, and basically we have no other choice to use them to understand what's going on there. What is, I think, also addressed by many of the people in the audience, link it to functions. And also the point is, yeah, can we really ever manipulate this in an experimental way? It's very complicated, but I think that's for sure also what has been discussed. This is an important next frontier, to really look at these networks, maybe measure them repeatedly, and then test them experimentally and see really, is this a keystone taxa that has a particular function? Is it important for restoring soil health? I think that Alison also mentioned that using networks for soil health prediction is still really not well understood. So there's a lot to do. I don't know if some of the other speakers, did I miss a conclusion? I think we slowly have to stop, actually. So maybe, Noah, you have some last words. Tancredi, Elly?
No, it sums it up really well.
All right, then I think we didn't answer all the questions. I'm very sorry for that. It was not possible, I think. But I think there will be other opportunities to discuss also in live in person at some time. So thank you very much, everyone, for your attention and interest in this webinar, and yeah, have a nice afternoon, evening, night, etc. Bye-bye.